Hi again, this is section 6 of chapter 3, where we're going to be talking about nucleic acids. This will be the quickest of the lessons because you kind of already know a good bit about nucleic acids. They carry genetic information, they make up DNA and RNA, and their monomer is a nucleotide. They serve an important role in protein synthesis. So they carry the instructions to build the protein. We have to copy the instructions on the mRNA. Our RNA makes up the ribosome. And of course, tRNA is essential in bringing the amino acids to the ribosome. So they're really important in carrying out the instructions found in our DNA. Polynucleotide is another word for DNA and RNA. It's like polysaccharide. The monomer is a nucleotide, and it's important that you understand the shape of the monomer. Now, on a very elementary level, this is a nucleotide where we have our 5-carbon sugar, which is either ribose or deoxyribose. And I'm just going to identify it as 5C sugar. Sorry, not closer. This is your phosphate. And this is your nitrogen base. We'll talk about those in just a second. And we also know the charge on the phosphate group, because you know this because you've studied your functional groups, is negative. So if DNA has a whole ton of nucleotides, each with one phosphate, the overall charge on DNA is going to be negative, which is great because it's soluble in water. But if I wanted to add another nucleotide, I'm going to draw it right up here. We've talked about the bond between sugars is glycosidic linkage. The bond between fats is ester linkage. The bond between nucleic acids, so when we're building the side of the ladder, we're going to bond between the phosphate of one and the sugar of the next nucleotide. And the name of that bond is phosphodiester. It is a phosphodiester bond that's in your PowerPoint, I think on the very last page. Do not confuse that with the bond that holds the ladders together. So if I were going to draw the other side, give me just a second because it's hard to draw upside down. Okay, so I'm going to draw my other phosphodiester bond between the sugar and the phosphate. So if this is a ladder and it were continuing to be built down the board, I would climb up the nitrogenous bases. The sides of the ladder are going to be stuck together, bonded with phosphodiester bonds between the phosphate and the sugar. So it would be sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate up the sides. You would climb up the nitrogenous bases, and the bond between these bases, you probably remember from Bio 1, is a hydrogen bond. It is that bond right there, okay? So don't confuse phosphodiester bonds, which hold the sides of the ladder together, with the hydrogen bonds that hold the middle of the ladder together. You probably also noticed that I had to draw one upside down. You will really understand why we do this when we go to DNA replication. It has to do with the orientation and the numbering of the sugar. This would be, or the carbon 1, 2, 3, 4, 5. We always have to build in the 5' to 3' direction. Okay, so this would be 5 to 3, and then that would be 5 to 3. So the two molecules are actually anti-parallel. They don't go in the same direction. They're actually opposite, anti-parallel. But we'll get to that when we do DNA replication because it has some pretty profound reasons or impacts on our chromosomes. Okay, this is probably new. Maybe you learned this in Bio 1, maybe not. You know the 4 bases, 5 if you include uracil, adenine, guanine, cysteine, thymine, and uracil. You need to know, stick it in your head, that adenine and guanine have 2 rings. They are purines. Pyrimidines are single rings. So if you take a look back at this, here's your elementary drawing of a nucleotide. And then you may see something like this on your quiz. Oh, that's a phosphate group. That's easy to remember. Oh, here's one single ring. That's got to be a sugar. Oh, that's got a bunch of ends, probably a nitrogenous base, and it has two rings. So I know that that's either adenine or guanine. I'm not going to have you memorize which is which. We know for a fact this is either adenine or guanine. So you just have to learn and memorize that A and G are purines and have two rings, and C, T, and U only have one single ring. All righty. I kind of talked about this numbering. You don't really need to know that just yet. You will need to know it later on, but you need to know the sugar is deoxyribose, and then it's ribose in RNA. Again, with the diagram, the purines have two rings, pyrimidines have one, and you can see that the outside of the molecule is phosphate sugar, phosphate sugar, phosphate sugar, phosphate sugar, all bonded by the phosphodiester bonds. And there's your phosphodiester bond right there. They're anti-parallel. They run opposite of each other. We'll talk about that later. Base pairing, you get it. Okay, and this is straight up from evolution. Two closely related species share more similar DNA than distantly related species. That is not earth-shattering and new information. If you have any questions about nucleic acids, hit me up. Otherwise, have a great rest of your day.